Okay, I'm going to take you through the first page of the cell transport cheat sheet. So try and follow along while I do this. You can also pause and play the video as needed to write down the stuff as you go. So we're going to start out with some definitions and kind of important information that you'll need to know before doing all of the rest of the types of transport. So as a reminder, selective permeability, the definition we're going to give here, some substances can cross, can cross the membrane more easily. Now, there are substances that have easy passage through the membrane and some that have a really hard time getting through the membrane or they need some assistance. So we're going to write these down. For easy passage, these are small, nonpolar, hydrophobic molecules. And our examples here are hydrocarbons and things like O2, CO2, N2, things like that. The difficult passage molecules are kind of the opposite. Large, polar, hydrophilic molecules and ions. Examples are sugars, which remember are carbs, and water, H2O. The next definition is for conformational change. We did talk about this in the last unit in regard to proteins, and it means the same thing here, but we're going to add a little statement that applies to transport. So a conformational change is a change in shape. And this next part isn't going to apply really until you start talking about pumps and like carrier proteins. But how it works is that ATP moves terminal phosphate to transport protein causing conformational change. So essentially what this means is, and you don't have to draw this, but if this is, here's the 3 phosphates in ATP. So this would be like the adenosine molecule, and then the little guys, the circles will be the phosphates. It's going to take this terminal, this last phosphate, and it's going to essentially donate it or move it to the transport protein, and that's what's going to cause those conformational changes. All right, and membrane potential is not something we're going to go into detail about today, but it will be when we talk about active transport. So it is an electrochemical gradient from unequal concentrations of ions across the membrane. The cytoplasm is relatively negative. Like I said, we'll talk about more about that one when we get to active transport. Now we're about to talk about passive transport as our first type of transport. And remember, you should have already had these notes done in your notes packet, and so this is going to be an opportunity for me to kind of share with you kind of what's going on with each one. Before I start getting into too much about it, let's talk about what a gradient is. Gradient means that there is more of something on one side than another. In art, gradient is like ombre, where it starts off darker on one side and the color moves lighter to the other. It just means that there's more of something on one side versus the other. And that's important information to know about transport, because we're going to be talking about how it moves either with the gradient or against the gradient. So passive transport, the first thing here, does not require energy from the cell. And it is responsible for import/export of materials. When looking at the gradient, passive transport is going to move with or down the gradient. And this means that it's going to move from high to low concentrations. So think of it kind of like a hill, right? And again, you don't have to draw this, but from high to low, if you run down a hill, it doesn't really take that much energy for you to do that versus going up the hill. So passive transport that does not require energy from the cell moves from a high to low concentration. Okay, diffusion. This is a spontaneous process due to constant motion of molecules. So because the molecules are moving all the time, diffusion is going to happen. Something you do need to know is that different molecules diffuse at different rates. And that's talking about the speed. So some molecules are going to pass through more easily, and so they may go at a higher rate, and then some are going to be a little bit more difficult, like water, to move through. And so it may take a little bit longer for it to pass through, so it'll have a lower rate of diffusion. And just like with passive transport, because this is a type, it moves with or down the gradient. That's high to low. Awesome. Okay, osmosis. This is specifically the diffusion of water. Now there are two options that you can go with osmosis about how things move. You can either look at the water concentration or you can look at the solute concentration. So it will move, water will move high to low water concentration, or you can say low to high solute concentration. And here's how a good way to remember it. Water loves solutes and will move to where there's more. So in osmosis, you're talking about water moving because it's the diffusion of water. So if you have a container, let's say here, and there's a higher concentration of water outside than there is inside. Water will move from high to low water concentration. However, if the opposite is true, and we have, so this would be for water, and you don't have to draw these. If for solute, if we have a high solute concentration outside and a low solute concentration inside, water will move from low to high. So it's going to follow wherever the more particles are. Okay, so we have here high to low water, I'm going to abbreviate concentration, or low to high solute concentration. All right, our last one is facilitated diffusion. The word facilitate means to help. So this is diffusion of molecules that kind of need some help. It's diffusion of, or diffusion with, transport proteins. There are two types of transport proteins that we're going to talk about. We have channel proteins, or carrier proteins. Facilitated diffusion so it increases rate of diffusion for small ions, water, and carbs. All right, so that we're gonna, in the next two boxes, we're gonna talk about the channel and the carrier proteins. So channel, for channel proteins, is hydrophilic. So the inside of the channel protein is gonna be hydrophilic, and that's what's gonna allow things like water and ions to move through, because those are also hydrophilic. Is sometimes gated. What it means when it says gated is that it's going to need or needs stimulus. Stimulus is something that stimulates. So it's some sort of charge or, kind of poking to do something. So sometimes the channels will be gated and something will have to happen in order for that channel to open. One of the most important type of channel proteins is an aquaporin, which is a channel for water. So when water needs help moving across the membrane, because it can diffuse through the membrane, it just takes a really long time. So sometimes the cell may need more water in or out, and so it may use a channel protein in order to help move water more quickly. So it'll increase the rate of diffusion. All right, for our next one, it is carrier proteins. And this is a conformational change. So the carrier proteins undergo a conformational change, or a change in shape, to let stuff pass. The molecules move. They still move. High to low. So carrier proteins are going to be the ones that kind of change shape, so they'll maybe open on the bottom and then change and open up on the top to let things in or out. Okay, this is your little cheat sheet to help you when we do stuff in class or study for your test, so that all the information about the types of transport is all in one place. Please don't forget to do the 5 practice questions from AP Classroom to help you with this content.